Life in its many forms, different sizes, shapes, habits, colors. Yet within this great variety there are strong patterns of order. The cells that make up each organism arise from previously existing cells. The process by which this happens is called mitosis. Cells are the building blocks of organisms, and they come in many shapes and sizes. A cell is not something simple. Each cell performs thousands of different and complex tasks. Like a chemical factory, the cell takes basic chemicals and from them produces new chemicals which are used by the organism and by the cell itself for growth and repair. In multicellular organisms, individual cells are specialized. Some perform muscle contraction. Others make up skin or transmit delicate nerve impulses. There are many kinds of cells with very different functions to perform, but all cells grow and maintain themselves. These are living human lung cells, shown in a unique side view. Every living cell is surrounded by a thin, flexible membrane that holds the cell together. Inside are structural and functional units of different shapes and sizes called organelles, which organize the complex chemical and physical work of the cell. The cell's activities are controlled from within its nucleus. Here, the instructions for performing all normal functions are issued, and here too are kept the special plans by which the cell replicates itself. Mitosis is the forming of two identical cells from the division of a single cell. Each of the new cells bears the same genetic information. The carriers of the genetic information are the chromosomes, located inside the nucleus. These are the chromosomes of a human being. There are 23 pairs, a total number of 46. This number is characteristic of our species. Each species of animal or plant has its own particular number of chromosomes, but the number has nothing to do with the size of the organism or how complicated it may appear. Fruit flies have only 4 pairs of chromosomes. Cats have 19 pairs. Some ferns have more than 300 pairs. A newly divided cell has a complete set of chromosomes which, at this stage, is not usually visible under the microscope. But with special stains, the nucleus is seen to contain dense tangles of threads. These threads, which become the chromosomes, are made up mostly of DNA, deoxyribonucleic acid, a long molecule that carries all the genetic information. As the cell develops, the DNA molecules split and the chromosomes are replicated. The cell now has 2 identical sets of chromosomes. The 2 identical copies are called chromatids and are joined at a point called a centromere. Mitosis can be thought of as an orderly series of events in which the pairs of chromatids are split apart and equally distributed to 2 new cells. Mitosis is one stage in the life of most cells. A cell enters a period of growth during which its size increases, and this period generally accounts for 30 to 40% of the cell's life. This is followed by a second stage of about the same length, in which growth is completed. The cell's DNA is replicated, and the various chemicals necessary for cell maintenance and replication are manufactured. The third stage is shorter than either of the first 2, but it is during this time that all chemical preparations for division are completed. At the end of this stage, the actual process of division, mitosis, begins. It is important to remember that the process of cell division is smooth and continuous. There is no stopping and starting between one phase and the next. Outside the cell's nucleus is a centriole. the centriole divides, and the two centrioles move apart. The nuclear membrane begins to disappear, and the thread-like nuclear material coils up into visible chromosomes. Many fibers connect the two centrioles, forming the spindle. The chromosomes line up in the center of the cell. The two chromatids of each chromosome separate and are pulled toward opposite centrioles. The cell divides. The chromosomes uncoil, and new nuclear membranes form. Here is the process of mitosis in a living plant cell. In living plant cells, the spindle is not apparent. The process of mitosis is continuous, but it is possible to identify four general phases, each characterized by specific activity. In the first phase, prophase, a centriole located outside the nucleus divides. The long thread-like material of the nucleus coils up into visible chromosomes, and the nuclear membrane disappears. From the centrioles, long thin strands extend in all directions. Many of these from one centriole join with strands from the other to form the spindle. The second phase of mitosis is metaphase, in which the chromosomes move into the equatorial plane of the spindle. As the third phase, anaphase, begins, the chromatids separate and move to opposite ends of the cell. Once the chromatids separate, they are called chromosomes. In this way, a complete set of chromosomes migrates toward each centriole. In the last phase, telophase, the cell divides. The changes now taking place are the reverse of those that occurred during prophase. The chromosomes uncoil. New membranes form around the nuclei, and the fibers of the spindle disappear. The cell has divided, and the two identical cells are now ready to begin their first period of growth. The actual process of mitosis can be seen very clearly as this plant cell divides. Here is the process of mitosis in a human lung cell, starting in late prophase. The process of mitosis is virtually the same for all forms of life. Mitosis is the basis for cloning. Cells that can trace their ancestry back to a single common cell are members of a clone. It is important to understand that cloning is a type of asexual reproduction in which, unless there is a mutation, all future cells will be similar to the first. One of the first successful clones from a single cell was done with yams. In the normal yam plant, stem cells, except at the growing tip, no longer divide, yet they have the capability for division. If the yam is wounded by cutting, cells will multiply and heal the wound. Then the cells will stop dividing. Some signal tells the cells to start or stop dividing. It has been found that certain substances can stimulate growth in these normally non-dividing stem cells. Groups of cells, however, will only grow so far, no more. In a group, each cell restricts those around it. But separate cells in culture can grow into normal adult yams. In the laboratory, pieces of stem are cultured. The cells are separated, and the individual cells continue to be cultured in nutrient solution. They are tumbled over and over in special flasks where they grow and divide, producing many new cells. These cells are passed through a series of hormones and then placed on agar, where they grow into new yam plants. Each cell in a plant or animal contains all the genetic information required to produce a whole new individual. Seedless oranges and many other kinds of fruits and vegetables are now routinely grown by grafting, a type of cloning. But what about animals? Is it possible to clone human beings? To take a few cells from an individual and make genetically identical copies? Already, nuclei from adult amphibian cells have been successfully transplanted into amphibian egg cells from which the nuclei have been removed. Through the normal process of mitosis, the eggs have divided and redivided to produce new adults, amphibian copies of the original. No one has yet cloned a new human being, but in theory, the recipe for mass-producing duplicates of a single human being would be similar to cloning plants and amphibians. There are many technical, ethical, and legal problems that may never be solved. In theory, though, the ultimate results would be infants who would grow up to be physical copies of the original person. Mitosis is the universal event in living matter. Multi-celled organisms, including humans, grow from one cell to many millions through mitosis, the orderly process of cell division.